right, ladies and gentlemen, we are going to jump into Chapter 5, Membrane Structure and Function. Now, 5.1 is going to specifically look at the membrane and how it is composed of phospholipids and protein molecules. Amphipathic means that these molecules are going to have both hydrophobic and hydrophilic regions. These phospholipids will have a hydrophilic head and two hydrophobic tails. Now, that makes the membrane regulate, or helps the membrane regulate what goes into and out of the cell. These blue portions here are the membrane proteins, and they can span the whole cell, or they can span just half the cell membrane here. And we're going to be talking more about that here in a little bit. Another cool image I like to show during this slide here. What they can do a technique called, it's like freeze cutting. I forget the specific name for this. What they do is they freeze a cell. They take a knife and they cut the cell and they cut the membrane in half. And you can see these images that they can get from the different sides of the membrane. It's inside of the cytoplasmic layer and the inside of the extracellular layer. Again, you can see kind of bumps on each, and this is just showing us what the membrane is made of. You can see that the tops here are the heads, and the bottom or the middle here are the tails. All right, the bends in the fatty acids of the phospholipids help with the fluidity. And I like showing this because the phospholipids are always jumping and moving around. Now, they're floating by each other on these sides. They can flip-flop sides, but it's very rare. But it does happen. But I just want to show you here that these membranes are made of phospholipids, and these phospholipid tails are very bendy, and they're always moving. It's called the fluid mosaic membrane. Now, these membranes are made of other things. You can see here they're made of a molecule called cholesterol. That cholesterol will add into the fluidity of the membrane. So more cholesterol, more fluid; less cholesterol, less fluid. And plants don't have that cholesterol. They don't need to be that fluid because, again, they're more rigid because of the cell wall. Glycolipids and glycoproteins. So these are two types of immune factors that are going to help your body recognize your cells versus not your cells. You can see here this glycoprotein. So we have glycoprotein, glycolipid. The glycoprotein is a set of sugar molecules that's attached to a protein. Glycolipids are a set of glucose molecules connected to a phospholipid. So again, glycoprotein, glycolipid. And these just help with the recognition of your cells versus other cells. This is how your immune system recognizes maybe a virus, maybe a bacteria cell is trying to infect you. If you know the ABO blood system from genetics, this is part of that. So one of the types of these glycoproteins and glycolipids is your ABO blood system. Integral versus peripheral proteins. So integral proteins go the whole way through. You can see A and B here are integral proteins. Then these blue ones here are the peripheral. They're either attached to a protein, they're attached to a lipid, they're on the surface. They can even be within this first layer, but they cannot span both. Those are integral proteins. So these are two main types of proteins that we're going to see in this chapter. Aquaporons, these are channels that allow water to pass through more easily. You can see the water passing through. If you remember from last slide, this would be an integral protein because it goes the whole way through. Now, water can pass this membrane, but it takes a little bit of time and energy because these are polar molecules, and we're going to learn that these polar molecules don't travel past the membrane very well. So they use these things called aquaporons. Now, these membranes are something called selectively permeable, which means they only allow certain things through. You can see charged ions don't make their way through. These water molecules have to go through those aquaporons. Uncharged molecules can easily make their way through, or somewhat easily. Larger macromolecules can't. So they need a different way to get in. And we're going to talk about these different ways that the cell is going to allow things in and push things out in this chapter. But understand that nonpolar molecules cross the membrane easily. Polar molecules, they need the help of a transport protein, which we're going to get to next. We're going to end on this meme here. Polar molecules, cell membrane doesn't like. Yep, nonpolar molecules. Yeah. All right, 5.2 is going to look at membrane proteins a little bit more in depth. Channel proteins, these are going to be membrane proteins, and these are going to be mostly integral proteins here. That these channel proteins allow for molecules to pass through. This goes with the concentration gradient. We're going to get more into that in the next section, but understand this just goes from high to low concentrations. This is just almost like a bridge that just allows cars to move past. It's just going to allow these molecules to move past that membrane. Carrier proteins. These are very similar to channel proteins, except in this case here, the carrier protein is going to change shape to allow these proteins pass. So very similar, but this one changes shape as these molecules move past. Yeah, there you go. All right, the next two, cell recognition proteins and receptor proteins. Cell recognition proteins are like the glycoproteins and glycolipids. They're going to be used to identify pathogens versus your own cells. And this is a very important type of protein because, again, this helps with our immune system. Next is going to be receptor proteins. These are going to be really important when we get into the next chapter. I know I say each one of these is really important, but they are really important. But these are going to be really important next chapter when we talk about cell communication. And basically what these do is they accept or they bond with certain molecules and they create some cellular response. This is pretty much how your body functions on a daily basis. If you're hungry, if you're tired, if you have whatever going on in your body needs to send signals throughout its whole body through the circulatory system. You have hormones, you have signaling molecules, and they connect with cells using these receptor proteins. So these are basically just locations where molecules are going to bind that just create some cellular response. And then the last two for this section, it's actually a very short section, are enzymatic proteins. We know a little bit about enzymes. They break down or build up molecules, but basically these types of protein, membrane proteins are going to be enzymes that are just fused within the membrane. Junction proteins, these will facilitate communication between two cells. You can see two membranes here, 1 and 2. But these allow for the passage, and basically these are like carrier or channel proteins that allow for the passage of molecules between cells, but they just conjoin kind of two cells together. This is all that there is for this section. It's a very short section, but I think it's really important that we kind of look at these a little bit more in depth because we're going to be talking about each one of these as we go on throughout the year. So again, receptor, you have molecules that bind that will create a response. Enzyme, they break down or build up molecules. We have channel proteins that just allow them through. We also have carrier, these gated channel, they're carrier proteins that change the shape to allow certain molecules through. Cell identifying marker membrane proteins, these identify cells. And then as you can see here, the CAM, these are just the junction proteins. So this is 5.2. All right, section 5.3 is going to be on passive transport. Concentration gradient. Areas of high concentration tend to move towards areas of low concentration. We talked about this a little bit in our lectures with 5.1 and 5.2, but basically what's going to happen here is things are going to move, or they have a tendency to move from high concentrations to low concentrations. And we can see the difference between the two here. Now, there are three types of passive transport. They are diffusion, osmosis, and facilitated diffusion. Now, diffusion, this is usually of like gases or molecules, but you can see in these animations here, they are moving from areas of high concentration to areas of low concentration. I'm going to go back and I'm going to see if it will go again. There we go. So you can see in both of these, they are starting out at a high concentration and equalizing by moving away from each other. And again, this is due to the characteristics of the molecules. They just don't want to be near each other. And again, they're moving from areas of high concentration to areas of low concentration. Okay, this is the first type of passive transport. Oh, interesting fact, this is how your body moves oxygen and carbon dioxide through your lungs and just the rest of your body. In your alveoli in your lungs, oxygen will move from areas of high concentration to low concentration to the red blood cells. And when the red blood cells go to your different organs, they will drop off that high the oxygen, there we go, and then it will pick up the carbon dioxide that your body has, and then it will deliver that carbon dioxide to your lungs. And this is all through diffusion, going from areas of high to low concentration. Osmosis, this is the movement of water, and it's usually through a semi-permeable membrane. And again, it's going from areas of high concentration to low concentrations. So you can see the movement of water from high to low, and it will equal out. Now, you can see there's this line here, that's that semi-permeable membrane. Even though the molecules can't cross, they will kind of help determine what is the high and low concentration. But again, we're looking at the water here, so the liquid. So this is just another type of passive transport. Ooh, let me go back. Like I said, yeah, molecules don't move across the membrane in osmosis. Now, there are three types of solutions when we're talking about osmosis. We have hypotonic, isotonic, and hypertonic. Hypotonic solutions. These are solutions that make whatever, like a cell or whatever we're looking at, swell, and they can actually burst sometimes if there's so much water going into the cell. I think hypotonic, I think that O, and think the cell gets bigger. Isotonic, there's an even amount of water and solute concentrations on the inside compared with the outside. So there will be an exchange of water, but it will be an even exchange. And then hypertonic, I think of like hyper as in like if you have a little brother or sister that's really hyperactive, I think small. So these are when the concentrations on the inside, we're talking about water, are higher than the outside, so the water moves out of the cell. So again, we're leaving the cell. It's hypertonic, so it's going to shrink. Let me show you what that looks like. Again, in isotonic solutions, they have an even amount of going in and out of the water because they're even amounts of the concentration. Hypotonic, the solution goes in because, again, we're going from high to low. And then hypertonic, water leaves, so the cell is going to shrink. And again, another example of this, you can see the hypotonic, the cell grows in size. The isotonic, it's an even amount. And the hypertonic, the water leaves, so it shrinks. Oh yeah, don't place salt on slugs. This kills them because it dehydrates them because they are it sucks out all the water because again it's going from areas of low to high high to low concentration, and the all the water in the slugs get sucked out. So we don't want to do this. Do not put salt on slugs. Plasmolysis. Let me go back. Plasmolysis. This is our last type of passive trans. Oh, I'm sorry. This is not our last type. This is a part of like the hypertonic solutions. Plasmolysis specifically is when the cytoplasm shrinks due to osmosis, and you can see this in the images or in these animations here, where the cytoplasm is shrinking. We're going to see this, I think, in lab this week. The last type of passive transport is facilitated diffusion. This is where there is still, you're going through the transport of high to low concentrations, but it's just through transport proteins like channel and carrier proteins. So the same thing, just across the membrane through proteins. So this ends 5.3. Let's look at these animations here. Yeah, again, transport proteins are pushing these molecules from areas of high concentration to low concentration. And we can see, again, more of these molecules going through the right channels. All right, so this is the end of 5.3. All right, our next section is going to be on 5.4. Here we go. 5.4 is going to look at something called active transport. And kind of like this name implies, active transport is going to be using energy. So we use energy in our cells in the form of ATP mostly. But active transport is just transporting molecules kind of against their concentration gradient. You can see here where we're going from a low to high concentration gradient. And there is a specific purpose for this that we're going to get into during photosynthesis and cellular respiration. But we're going to see this whole process again later on in the year. So you can see just these transport proteins are going to push molecules into a smaller area, and we're going to raise the concentration. All right, the first type of active transport we're going to talk about is exocytosis. Exocytosis, you can see here, is when these vesicles fuse and they push something out. We saw this back with the endomembrane system, the cells producing proteins that go outside the cell. These are extracellular proteins, but we also have a lot of other things or a lot of other processes that include this exocytosis. So think exo, exit, and you're exiting the cell. Endocytosis. You can probably guess what this looks like or what this is. This is when the cell takes in molecules, and then you can see it wraps its vesicles around it, that plasma membrane. So you can see here endo thinking in, wrapping the membrane around whatever it's taking in. There you go. Video here we're going to skip. All right. Two types of endocytosis that we're going to get more in depth with. Phagocytosis and pinocytosis. I say this one just a little bit weird. Just makes it, we're just going to say it a little bit weird. Occurs when the cell takes in large substances. You can see like a molecule being taken in here, like a solid molecule. And then pinocytosis. This is when a cell takes in really small substances and mostly liquids. This is how bacteria actually kind of drink. I know it's weird thinking bacteria drinking, but they need liquids. So that what they're going to do is they're going to take in small vesicles of liquid, and they just incorporate that liquid into their cell. Receptor-mediated endocytosis. So we talked a little bit about these receptor molecules, the receptor proteins that will bond with something. What happens then is other molecules will also sometimes come in, but these molecules then will trigger some type of endocytosis and go into the cell. So this is another type of active transport, getting things into the cell. Oh, yeah. We talked about how your immune system does this. We talked about how when the bacteria infect the cell, a lysosome can bond with it. So this is both endocytosis and exocytosis. All right, 5.5. This is going to look at a little bit more in depth with the extracellular matrix and the other membrane connections between cells. So we talked about the different membrane proteins. Now we're going to talk about how they can kind of create a barrier between two cells or create some connection between two cells. The next chapter after this is cell communication. So this deals right along with cell communication. But anyways, back to here. The extracellular matrix, this is a meshwork of like proteins and carbohydrates that are located right outside the cell, and they hold the cells together and they help communicate signals between the cells. The green stuff in this image here you can see is the extracellular matrix. You can see it's connected by the cytoskeleton through the transmembrane proteins. So you can see the cytoskeleton beneath here. You can see the collagen fibers on the outside, and they're connected by these transmembrane or integral proteins. Holds tissues together, yes. Okay. Collagen, it's the most abundant protein that you're going to find in your body. Again, it gives that kind of meshwork or just a framework for your cells to bond together to form these tissues. Another way of looking at it, you have your collagen, you have your cytoskeleton. They are fused together through these membrane proteins. All right, desmosomes. I always like to call these spot welds, and then I realized that not everyone knows what a spot weld is. Basically, a spot weld is when you take two sheets of metal and you press them together, and then you weld just like a spot, and it holds those two sheets together. Basically, the same thing here with the cell. These desmosomes are membrane proteins, and the kind of proteins that go between, you can see here, they hold cells together. So these are just like places where you're going to glue these cells together. And again, this is what holds the cells together to make tissues, make larger structures. I love this meme. Good. Cell, desmosomes. Yeah, they hold cells together. Tight junctions. These are also membrane let me go back. These are also junction proteins where you're going to see the almost like a line produced, but it's like a watertight line surface, or it's a watertight surface where water's not going to be able to pass. Excuse me, across these membranes. Again, these are just places where the cells are going to fuse together. That's basically all. These are junction proteins. Yeah. And lastly, we have these gap junctions. Gap junctions are a tunnel. We talked about these before. They're like a tunnel between the two cells, and it helps with communication. We do want to mention plasmodesmata here. These are narrow channels that are located in plant cells. Remember, plant cells also have that cell wall. So how do they get their communication or their signals between cells? They have a surface, not a surface. They have a hole between the cells that allows that passage. Because again, if you just have the cell membrane, it's easy, but adding the cell wall makes it a little bit more difficult. But they are junctions between the plant cells called plasmodesmata. So this looks like the end of the chapter. Yeah, chapter 5 is done.